everyone. My name is Lili Chan. I am currently a faculty of nephrology at Mount Siani in New York, and I wanted to welcome you to the Q&A session of Dr. Marcus Pereira, who is an infectious disease specialist at Columbia University, who gave a talk about updates on COVID-19 vaccines, and also Dr. Alp Ikizler from the Division of Nephrology from Vanderbilt, who spoke about COVID-19 vaccinations in kidney patients. Please, if you have any questions for either of our panelists, put them into the chat or the Q&A function. I'm going to go ahead and start with one of my questions is Dr. Ikizler and also Dr. Pereira, you both spoke about how CKD patients, dialysis patients, and transplant patients weren't included in any of these initial trials. So how do we convince our patients who may already be hesitant to get this vaccine that they should be getting it?
Marcus, you want to go ahead and give it a shot first, and then I'll
sure
have my thoughts.
Sure, not a problem. Thank you, Lili, and thank you for the organizers for allowing me to talk and for this Q&A session. I'll speak a little bit about the transplant patients, and I'll let Dr. Ikizler talk about hemodialysis and patients with CKD. From a transplant perspective, it certainly was a major gap in the initial studies that those patients were not included in those studies. So really the knowledge was very limited going into the approval of these vaccines and the rollout in terms of how they would function in the immunocompromised patient population. So we were concerned based on historical precedents from pneumococcal and influenza vaccines that those patients would have a reduced response, and also on some preliminary studies about reduced antibody responses from natural infection due to SARS-CoV-2 throughout 2020 that were reported. So there was certainly a concern, and then preliminary studies at this point do seem to suggest that there might be a reduced rate of antibody response after the mRNA vaccines in particular. That's the data that we have, and I think there's a number of emerging publications pointing to that. But it's all preliminary data, and it's only just serological data at this point, and we don't know necessarily what the significance of those results are in terms of protection from either infection or severity of disease. I imagine we'll be talking a little bit more about that in sort of deeper details, but I'll let Dr. Ikizler just give his initial thoughts on that as well.
Yeah, Lili, thank you very much for allowing me to participate in this, and I'll put this up front for the people who listen. I'm not an immunologist, and over the last 3 to 6 months, I've learned more than I've ever learned in medical school about vaccines and immunizations, and not only for this talk, but also for our own general public knowledge and what vaccines mean, how we really assess vaccinations and everything. And one thing that I've actually come to learn is that we know very little. And I think that sort of goes back to your initial question, that is, we have not been able to get the dialysis patients or transplant patients or even stage 3 and 4 and 5 CKD patients into the trials, where we really learn a lot. And again, one thing that one can actually take away from this, for the listeners, this is true for all vaccines. This is not only for COVID. And people just sort of feel like it's being done for COVID vaccination, but that's not true. We have had very limited information about influenza vaccines. We have very limited information about vaccination rates and immune response in solid organ transplants, including kidney. And you would think we should know that. And we're still struggling with Hep B vaccinations. So everything put aside, I think we have a problem that we have to solve. And I go back to my good colleague, Dr. Glenn Chertov's and others' comment about renalism, a nihilistic approach to kidney disease patients, that these patients should be avoided from any kind of early trials because they're a high-risk patient population. One thing that we can take away from the knowledge we gain from COVID is that that's not true. Actually, the vaccination seems to be pretty safe in kidney disease patients. So it's not like, you're taking a group of patients who are going to have major problems in terms of new vaccines and heavy issues. So this concept of we shouldn't be including them in the clinical trials is not true, is not correct. And if you look at that paper that I've shown you from UNC about the clinical trial inclusion, we need to be a little bit more aggressive. And to make it, just to wrap it up, I will tell you we've tried through ASN, through NKF, through COVID-19 response team, through Kidney Health Initiative, where we had connections with companies to push for dialysis patients to be included in these vaccine trials. And had it not been this warp speed vaccination process, we probably would have succeeded and added some patients into the early trials. But everything was moving in a light speed in terms of getting the information, getting the studies done, and getting the information available to the public. So there's a good and bad that's attached to this COVID vaccination process that we learned a lot. We know what to do next. And I think we will be able to convince them. And that what I would say to your second question, that is how we convince our patients to get the vaccine, we just tell them it's safe. You know, we learned that it is actually safe for these vaccines. A lot of them, in my own personal interactions, are concerned because they've given that unreal nihilistic approach that you're a very vulnerable patient population and maybe you shouldn't be getting this. You should be very careful, but that's not true for these vaccines. The safety profile is pretty good. If you look at the available data that's been dumped into the literature over the last week or so, dialysis patients are doing as good as any other patient population or healthy population in terms of the vaccine effects. Sorry, it's a long-winded response, but I think that's what I learned when I did this reading and understanding of the vaccine issues in dialysis patients and transplant.
And this actually ties in very nicely to one of our audience questions about, with the hesitancy of vaccination. Do you have a sense of what vaccination rate needs to be obtained for herd immunity?
I'll leave it to Marcus. I have my thoughts, but it'll be totally a guess. But he's an ID person. He should answer that, and I'll learn from him.
Well, I'm going to disappoint you because I don't think anyone knows. There have been a lot of guesses around, but I don't think that there is a magic number. Obviously, people are talking about 80%, 70%. You can look at certain countries, for example, like Israel, which have been sort of at the forefront of vaccination efforts. And it clearly seems to have impacted the rate of infections overall in the country. Now, I am not sure if we know exactly at what point in terms of the total percentage of the population that has been vaccinated, in which it made a population-wide impact. But you can imagine that, the risk is also it needs to be individualized to how much risk one takes on a daily basis due to work, due to travel, due to other exposures. So it really, it's a lot more complex than sort of one number, whether it's 70, 80, or 95% vaccination. And remember that we're nowhere near those numbers, beginning with the fact that the pediatric population has not been vaccinated, right? So that's already 25% of, for example, the US population. And then you factor in even the sort of the patients who are at high risk of severe COVID-19, sort of those over age 60, we're still not sort of at a significant levels of vaccination. So I don't think that even focusing on that necessarily is an important sort of aspect of this. I think it has to be put together with ongoing safety practices like wearing masks, safe distance, social distancing, and other practices, along with the vaccination. I think then at that point, I think that we're talking about some measure of herd immunity or populational control over this.
Can I add a follow-up to that, to Marcus, or my own knowledge? How do you combine the herd immunity that you gain from the novel infection plus the vaccination? So do they sort of overlap with each other? And can you do your counting like 1 + 1 = 2?
Wow.
20%, like in New York, a substantial portion of people got the disease. And then you vaccinate the people who didn't get the disease. Do you add them up as part of the herd immunity?
That's a fantastic question. And actually segues to another thought that I did not mention. It does not add up 1 + 1 = 2. And one of the reasons why we're really concerned relates to these variants, in that potential immunity to the original strain of SARS-CoV-2 may not render one immune to the subsequent variants. And one of the cases that I point out during my talk was in the city in Brazil named Manaus, which is in the Amazon. They had significant rates of infection in April of 2020 with the original strain. And by some accounts, more than 75% of the population had antibodies to SARS-CoV-2 when they were doing sort of plasma and blood donation investigations. And this was in the late summer. And what happened in Manaus in their summertime, which is in the Southern Hemisphere, so around December, was that cases started picking up again. And in fact, actually, it was a recurrence of what happened in April in terms of high numbers of patients getting infected, many with severe COVID-19 symptoms requiring hospitalization. And they actually had almost a breakdown in their healthcare system in terms of oxygen supplies and other things. And really that was sort of the an indication that, for example, the Brazilian variant, the P1, was breaking through prior immunity. So natural immunity actually may not be, for example, enough protective against the variants. What we don't know is the impact of vaccination, whether that is more potent than natural infection. So some of the preliminary studies have shown that those who are vaccinated, their antibodies might be somewhat neutralizing against the South African, the UK, and the Brazilian variant, perhaps a little bit at a lower level than the initial SARS-CoV-2 strain, but still thinking that it's protective enough. So in fact, in many ways, I think that this goes along with the sort of general thoughts that, vaccinations, because they're so targeted in their immune response, in some ways can be more immunizing or protective than natural infection. And that's what we're hoping with these vaccines. So the real hope is vaccines, not necessarily natural infection in terms of herd immunity, to sum that up.
Excellent. Yeah.
So I think, Dr. Pereira, in your talk, you talked about the development of the vaccines and how most of the vaccines are targeted against the spike protein currently. And the variants do have mutations in the spike protein currently. So are there any vaccines in development that don't involve the spike protein, or is there any other potential targets there?
No, that's an excellent question. We really did put all of our eggs in that one basket of the spike protein. I am not aware of different targets. There might be one or two, certainly not in the United States or Europe, that have looked at, for example, the nuclear capsid or tegument or other sites. The bottom line is, it really is that the spike protein is the most abundant sort of surface antigen in the virus, and really is the prime target for immunity. So it made a lot of sense that all of the vaccine developers really focus on that antigen. And it seems to have paid off, right? I mean, we were incredibly lucky that the initial sort of results from these vaccines were beyond our dreams in terms of 95, 93% efficacy from the mRNA vaccines. But as you point out, the spike protein, the caveat is that it does mutate a lot, and those variants really, this is where they're mutating. It's at the receptor binding domain of the spike protein. So some of the antibodies produced by the vaccines are effective enough to neutralize these variants, whereas others may not be. And this is where we still don't know how these vaccines will protect us against these variants. The case in point is, for example, that the Oxford vaccine really had really disappointing results in South Africa with only about 10 to 20% efficacy. The Novavax and the Johnson & Johnson did a little bit better. And then some data from the mRNA vaccines regarding the South African strain, for example, are still very preliminary and in vitro only, but those seem to be a little bit better. But the key is going to be how this virus mutates in the subsequent months and how we're able to still control it with these vaccines.
I was just going to add one thing, and this is not because I know and follow more, but it was published by investigators at Vanderbilt that there's in Cell very recently, that there are some monoclonal antibodies that have been produced against the N-terminal domain, which they suggest that those specific targets can actually be placed into the new vaccines because they're more protective than the spike protein and they don't really mutate as much.
And that would be a smart strategy, right? To focus on multiple different targets, not just the spike protein, but sort of have a combination of targets, whether it's in the vaccine or with these monoclonal antibodies, so that you don't allow the virus to escape. We have a model with that with combination therapy against HIV, for example, and other viruses. So it really does make sense to do that.
So we have several questions asking, do you think we're going to need vaccines every year? How do we handle these variants? Are we going to need boosters? And even specifically for our dialysis patients and our transplant patients who may not mount the same antibody response as the general population, like, do we have to treat them differently than everyone else?
I'll let you go first, Dr.
Okay, I'll just give my very limited knowledge, which I think is crucial. And I was informed by one of my colleagues in France that the French health authorities have allowed the third vaccination for dialysis patients, transplant patients, and immunosuppressed individuals 90 days after the first second vaccine, or whatever the 90 days. I don't know the exact date, but they've come to conclusion that there's going to be a need for a booster. That's all I know. And that's all I'm going to say. And I'll let Dr. Pereira just sort of give the real data. But there seems to be some enthusiasm to do that.
Yeah, I don't have any inside knowledge, but I do have my strong suspicion that third or subsequent injections will be required, perhaps in some particular populations, like the immunocompromised individuals who may or not be responding to the sort of two-dose vaccine series. I think there's a lot of still a lot of gaps that remain to be filled in terms of our knowledge. And this ties in with our initial question, right? It's sort of these serological studies. Are they sufficient to sort of give us a clear picture how well patients are responding, or whether we need sort of deeper analyses like T cell reaction or reactivity to these injections? And I suspect that we need that data to really complete sort of our understanding and how well someone is responding to the vaccine, and perhaps even create some form of score or analysis on whether someone has responded and will need subsequent injections. Much in the same way we do with hepatitis B in measuring surface antibodies after the vaccine series and determining whether they need further injections until they form sufficient surface antibodies. I think that motto is there, and I think we'll probably follow that as soon as we understand what the correlates of immunity are. Now, for those who respond, I think that there are long-term studies ongoing, 6 months, 12 months out from the initial vaccine, and probably we'll be measuring those same correlates, whether it's the spike protein, IgG alone, or some form of T cell analysis from CD4, CD8 cell responses that are specific to spike protein, for example. And based on those results, then determining whether those who have immune competent systems, will need a third injection. The other question is whether the variants themselves will sort of push us towards adopting the vaccine, which can be easily done with the mRNA vaccines, for example, and even sort of the adenovirus vaccines that actually can be easily sort of altered to adapt to those variants and then give subsequent injections. I suspect that we'll all be getting further shots.
So out of curiosity, just to make sure, although it's not applicable to the United States, but there are certain countries that are using the live attenuated vaccine, like the Chinese vaccine and others, so they will not be able to switch for variants. Isn't that correct?
Yes. Yeah. So that would be much more difficult. Absolutely. That seems sort of an older platform that we have used, right? So the measles, mumps, rubella vaccine, for example, those are live attenuated. That's incredibly hard to create a new vaccine on that platform. The same with the Novavax vaccine, which is protein based. Again, sort of an older model, right? Much like how our pneumococcal vaccines are. Those are not necessarily very easy to create because chemically, it really depends whether the spike protein is stable chemically in the vial and whether those variant spike proteins are stable as well. So this model of mRNA and sort of viral vector DNA vaccines, it really is sort of revolutionary in that way because you can easily adapt to changing circumstances and roll out very quickly novel forms of the vaccine. So it's really one of the silver linings of this whole pandemic, for sure.
Given that there's so many different vaccines available and the need for likely a booster or additional shots, do you think we'll have to keep track of which person gets which vaccine? Will it have to be a booster dependent on what they previously received?
Well, that's a good question. So some people have toyed with the idea of switching vaccines, perhaps for enhanced immunity. It seems that it's safe. I know that there are a couple of phase 3 trials with the Oxford vaccine and some of the mRNA vaccines that we're undergoing and sponsored. So it seems that it's safe. Now, whether it's more potent that way versus sticking with the same type of vaccine, I think that remains to be seen. It's certainly interesting and would make sense to sort of hit the immune system with sort of different platforms so that it's not sort of, not giving something novel for the immune system to adapt to.
This might be a question for Dr. Ikizler. There's been a number of adverse events now reported, including vaccine-associated thrombocytopenia. Are our patients who are on dialysis who receive heparin at an increased risk?
Okay, so this is a very hard question. I'll let Marcus also participate in this. This is an evolving piece of information. I've been trying to keep track of what the information and what's relevant to. What I understand right now, it's the adenovirus vector that seems to be implied in this whole process, initiating an immune response, subsequently causing something else. But usually the recommendations that I read is that not to treat this problem with heparin, rather than heparin itself causing a predisposition to the problem to begin with. So when this happens, ITP-like picture happens, you don't treat people with heparin. So I would say, based on my limited knowledge and speculative approach, no, it should not be a risk. But if somebody develops that rare entity, which seems to be very limited to a certain group of individuals, I'm not even going to say a group in general, but just a very limited number of cases, it does not seem to be a problem to have being treated with heparin previously, is my take. I don't know what Mark's got to say.
I agree with Alp. I think overall, sort of this vaccine-induced thrombotic thrombocytopenia seems to be correlated with the adenovirus vaccines. Right, first there was sort of these news from the Oxford vaccine in Europe that they were reporting some unusual cases of thrombotic events, sort of not the regular deep vein thrombosis cases, but sort of things like splenic vein and portal vein thrombosis. And then the ones who made more of the news, right, the cerebral venous sinus thrombosis events. Those seem to be rare enough, and soon enough the vaccine was sort of re-released in the European market. And then came the Johnson & Johnson vaccine, similar vector, right, the adenovirus, although a different adenovirus, sort of similar platform, and now we're seeing cases of that as well. On the whole, it seems that these are very rare. These were, for example, in the United States, there were 6 reported cases out of more than 7 million doses given. So you can imagine sort of one in more than 1 million cases from this vaccine. And this compares to sort of other more common risks, like even oral contraceptive use, that risk seems even higher than that. Some of the case fatalities that came from these episodes, exactly what Dr. Ikizler was alluding to, that these were sort of more on the spectrum of heparin-induced thrombocytopenia, in the sense that these patients had this platelet factor 4 production and elevated levels of that, and they were also thrombotic. So it was clearly something sort of unusual in the sort of clotting cascade that was occurring, perhaps sort of an immune related process. And it seems that obviously using heparin seems the wrong thing to do. So what we're hoping is that sort of once we fully understand what are the risk factors for developing this rare event related to these vaccines would be, whether it's age, gender, certain characteristics, those all remain to be seen and are actively studied right now by the CDC and other agencies. I think that then we can sort of either tailor the vaccine not for those recipients, or at least have sort of enough of a warning that should they develop symptoms consistent with some of these rare clotting, know how to manage it effectively and rapidly, i.e. not give heparin, give alternatives in terms of anticoagulation, and diagnosing those soon, because those are manageable problems. And certainly you can avoid further deaths now that we sort of understand what this risk is. And I think the most important message that we can give our patients in the community is that, on balance, the risk of dying from COVID-19 or developing severe COVID-19 is far greater than of these rare events, even from the Johnson & Johnson vaccine. So I think that once it's released and we better understand, these ought to be still recommended for our patients and actively taken, because this is our way out of the pandemic, and we really need to be very focused on that.
Do you think when the Johnson & Johnson does become available again in the United States that we should be using it in our dialysis patients if they're at risk for clotting potentially of their dialysis access? And also based off of the studies that Dr. Ikizler showed us, that we really don't see an immune response in our dialysis patients until after the second dose. You know, is that unique to the mRNA, or is that also going to extrapolate out to the J&J, which is only one dose?
I'm going to ask Marcus this. What I figured out is that the reason why J&J picked one dose is not because works that way. It's just they've decided to, from the get-go, without a specific reason. So my guess is that it takes about 3 to 6 weeks to mount a immune response. And, you know, this goes back to my initial comment. I'm going to repeat this in the setting of 40-something attendees. Our knowledge, at least I would say, I will take myself and put it on the spot and say, my knowledge of vaccines in dialysis patients in general was very limited. This has led us to learn a lot. And it takes a certain period of time for the vaccines to work. So I don't think it's a 1 dose or 2 dose, just you got to wait to see your real response. Is that a reasonable statement from a naive individual in vaccination, Marcus?
Well, I wouldn't put you in that category. I would put you in the very knowledgeable category. And I think you're absolutely correct. Vaccines take time to develop an immune response. I think expecting them to be fully effective within 7 to 14 days, I think it's definitely unrealistic, or even 21 days for someone to develop fully active, antibodies. So, and that's why all of these initial studies are very preliminary. And we really need to understand what that sort of translates into actual immunity and protection. For example, the Johnson & Johnson vaccine, if you look at its efficacy and sort of that curve, that control placebo versus vaccine arms, the efficacy of the vaccine actually increases with time. By day 45, that's when you see 80% efficacy. And it really is a function of that 1 dose of the vaccine that they gambled. Like you said, I think that they felt very confident in their vaccine and gambled in their one-shot approach, and it paid off tremendously. I probably imagine that Oxford probably wishes it had done that as well. But you're absolutely right. I think it takes time for these vaccines to actually have their full effect.
I think we're in our final minute, so I'm going to do one last question. And this is from the audience. Has there been any evidence or concern for increased antibody-mediated disease enhancement with vaccination? Would the blood clots developing be evidence of this?
I can be very quick. To my reading, although the number of publications in COVID has surpassed my limit of reading anything, I have not been able to find that. There's no documentation. What I've learned over time is that it takes time to really develop an understanding of vaccine complications, because unless it's a very obvious big event, it accumulates over time. So at this point, I have not seen, but there may be some that people might be reporting over time through, pharma databases.
And especially for transplant patients where they're at risk for rejection, is that a concern for you, Dr. Pereira?
It had been initially a concern. We have not seen any signal whatsoever that patients who are vaccinated with any of the vaccines are developing higher rates of rejection. However, like Dr. Ikizler said, it's probably too soon to say, 1 to 2 months after vaccine. I think we need more longitudinal data to really establish that these are not risks. But given all the history of prior vaccinations and prior concerns with numerous different platforms, it does not seem that vaccines can lead to rejection, antibody or cellular mediated rejection. I think it's safe.
Great. So we are actually at the hour, and I want to thank you both Dr. Ikizler and Dr. Pereira for a great panel discussion, and also everyone who joined us. Unfortunately, we weren't able to get through all of the questions, but I hope everyone enjoyed the session.
Thank you. Thanks for the opportunity.